Hi everyone, I'm Katie and I'm going to be talking to you about how macrophages and circadian clocks affect homeostasis of collagen. And I'm in Dr. Joan Chang's lab, co-supervised by Carl Cadler, Tracy Hussle and Mark Travis. So we'll start with this maybe vaguely gruesome image. So on the left we have the human lung and on the right we have the decellularized human lung. So that's just the same lung as on the left but with all the cells taken out of it. And as you can see, it's the same approximate size and shape, but all we're seeing on the right is the extracellular matrix. So that kind of gives you an impression of how important the extracellular matrix is to surround our cells and our tissues and our organs to allow them to fulfill their functional properties. And the predominant and, in my biased opinion, most important protein in the extracellular matrix is collagen. And collagen is found in skin, bones, ligaments, and it's especially important in load-bearing tissues, including muscle, cartilage, and tendons. And like lots of things in our bodies, collagen needs to be carefully regulated, and too much collagen can lead to fibrosis. So if we look at the diagram, the cartoon on the right, so on one side is a healthy lung, and this would be regulated collagen deposition, and it would allow our lung to perform efficient gas exchange. But on the right, there's a fibrotic lung, so this would be unregulated collagen deposition, and this would lead to tissue which is too thick and inelastic, and in the lung, this would lead to decreased gas exchange. So fibrosis is a really important and prevalent condition. So nearly 45% of all deaths in the world can be attributed to fibrosis. And as of yet, there are no effective treatments for the dysregulation of collagen. And this is in part due to the fact that we don't really understand how collagen is regulated and even less about how it's what drives its dysregulation. And that's what my project is looking at. So how does collagen work when it's being regulated and doing its job properly? So if we use an example of if you had a cut or an injury in your skin, that would be an epithelial injury, and what would happen then is fibroblasts would be activated and recruited to the site of injury. They would deposit collagen and remodel the ECM, and this would lead to nice resolution and no scar tissue. But in a scar, the work done by Paul Riley down in Oxford has shown that in scars we can see macrophages rather than fibroblasts being the predominant collagen-producing cell. And scars are similar to fibrosis, so this is kind of the work we've been kind of leading on from. So this kind of led us to suggest that macrophages and fibroblasts may be having some sort of crosstalk which leads to incorrect collagen production, leading to fibrosis and scars. So yeah, that's kind of what I said. So the macrophages recruiting and incorrectly producing collagen, leading to fibrosis. So yeah, the hypothesis is that the ECM remodeling and collagen deposition relies on crosstalk between fibroblasts and macrophages, and these are the two predominant collagen-producing cells. And potentially when this crosstalk goes wrong, we see chronic scarring and fibrosis. And this is the question that I'm going to be trying to ask. What is driving this dysregulation of collagen and fibrosis? So yeah, as I mentioned, macrophages have been shown to produce collagen in wound sites, but they haven't been shown to produce collagen on their own without any other external stimuli from cells or other factors. So that was the first step and quite easy experiment to show that macrophages themselves can deposit collagen fibrils. So I polarized macrophages into M0, M1, and M2. And in green, we can see the collagen and the kind of continuous lines are actually fibrils. So yeah, that's just a kind of first pass experiment to show macrophages can deposit collagen fibrils without external stimuli as they would do when they're in wounds in a mouse. Secondly, I wanted to know how fibroblasts and macrophages actually interact and whether one can affect the other in terms of collagen deposition. So on the left, we have the fibroblasts on their own, and you can see collagen being deposited, but no fibrils forming at this point. And then in a co-culture of fibroblasts and macrophages, you can quite clearly see that there is an increased deposition of the collagen fibrils. And you might be saying, I've just told you that macrophages themselves can produce collagen fibrils, so how do you know this is coming from the fibroblasts? And that's because the fibroblasts in this experiment are from mice and the macrophages are from human, which isn't ideal, but what it does show is that the collagen is from the fibroblast because macrophages need a cofactor, they need ascorbic acid to produce collagen, and we didn't use the ascorbic acid in this experiment, so we can be fairly sure that it's from the fibroblasts. So that's our second finding. So there is a crosstalk between macrophages and fibroblasts which increase collagen fibril deposition by fibroblasts. So we're going to switch gears a bit now, because as well as the involvement of macrophages in collagen regulation, it's also thought that the circadian clocks may have an effect on collagen synthesis. So for those of you who aren't familiar with circadian clock work, so circadian clocks are our body's natural timing device, and it regulates our physical, mental, and biological changes throughout a 24-hour period. And we have 4 main clock genes shown on the screen, so BMAL, clock, pair, and cry. And they act as transcription factors to regulate expression of clock-controlled genes. And it's been shown that some genes responsible for collagen synthesis are actually clock-controlled. So it kind of therefore follows on that perturbations in clock genes in fibroblasts could change how and when collagen is deposited. So to look at this theory, we wanted to measure the circadian rhythms in fibroblasts and then add macrophages on to see what would happen to the rhythm. So to do this, we use the PER2-LUC mouse. So the PER2 mouse has a PER2 gene, which I just told you is one of the clock genes, and it's fused to a luciferase gene. And when we add luciferin in, which is its substrate, we can measure the luciferase signal, and if we see a nice rhythm in the luciferase signal, that reflects that there is a nice robust clock in the fibroblasts. And then we can add the immune cells and observe what happens to the rhythm in the fibroblasts. And going back, this all relates to collagen because, as I told you, the fibroblasts are the main collagen-producing cell. So we are seeing if the circadian rhythm changes in the fibroblast will affect collagen deposition. So here's some data from the fibroblasts, the PER2-LUC fibroblasts. So the red trace is just the PER2-LUC fibroblasts on their own with no rhythm. So you can see it kind of flatlines out after the initial first peak. And then in the blue is where we've added dexamethasone, which is a corticosteroid used for synchronization in lots of different assays. As you can see, there's a nice big peak and it carries on for around 4 days. So that's showing there's a nice robust rhythm. And then in orange is where we add macrophages. And although it's not as high or in amplitude as the dexamethasone, you can still see the orange trace has a circadian rhythm. So yeah, that kind of just showed that the macrophages are somehow inducing a rhythm in the fibroblasts. And we guessed that this could either be by cell-cell contact or by a cell-secreted factor. And to work out which one of those it was, we first looked at whether it was a cell-secreted factor by taking cultured media from the macrophages and spinning it through a 50 kilodalton filter to get 2 fractions. So the substances in the CM which are greater than 50 kilodaltons and those which are smaller than 50 kilodaltons. And we also heat treated the CM, so we boiled it for 10 minutes, and then we added all these different fractions to the PER2-LUC fibroblasts. And basically what we saw was that the fractions containing components of the CM which were greater than 50 kilodaltons produced a rhythm, so that's the dark blue trace. Things that in the smaller fraction, so things that did pass through the 50 kilodalton filter couldn't induce a rhythm, so that's the purple trace, which kind of flatlines. And interestingly and surprisingly, the heat-treated CM in orange also produced a rhythm. So this kind of tells us that the induction of circadian rhythm in fibroblasts is driven by a heat-resistant macrophage-secreted factor, which is larger than 50 kilodaltons. So yeah, that's, as I said, and this is kind of what my next steps will be to try and work out what this factor is. And to do this, I'm going to use a larger filter, just a very crude way of determining whether the factor is between 50 kilodaltons and 100 kilodaltons. I'll then use size exclusion chromatography to separate the components of the CM and then add them to the fibroblast to see which fraction can induce a rhythm, and then identify the different size components within the CM using LCMS to work out their possible identity. And finally, just to acknowledge the whole of the CADA lab, in particular Joan Chang, Marie and Tracy Hussle, and the SLOLA group, which we have a collaboration with down in Bristol, in particularly King John. Thank you.
Another lovely, clear talk, thank you. So we have time for some questions. Judy?
It's really nice to see the macrophages making the collagen fibrils. Were they able to do it? I mean, maybe you said and I missed this. Were they able to just do that on their own without any sort of external stimulus? They were just making collagen?
Yeah, so we have to add ascorbic acid, as I mentioned, because that's a cofactor to make the collagen from the procollagen. But apart from that, they were on their own. The only thing I would say is it is quite tricky. This conditions need to be quite precise, so they need to be at very high confluency. And they need at least kind of 5 to 6 days before you start seeing any fibrils. Before that, it's just kind of like fragmented spots where they are releasing collagen but not forming fibrils.
And was there any, I mean, I don't know if you had a chance to quantitate really, but you didn't show any difference between the different M0, M1, M2, but is there?
No. So I've quantified that with ImageJ, but it seems that there's no obvious difference.
Question from Andy.
Hi, thanks. Really interesting. Do you know if it's cell specific to macrophages? Have you tried it with endothelial cells or other like smooth muscle cells, or just any type of the cell type?
For the rhythm.
For the rhythm or the extra collagen deposition.
I haven't for the collagen deposition. That's a really good point. But for the rhythm, it looks like I've tried different macrophages, so cell line and from mice, and it seems that all macrophages can induce a rhythm. And it seems that particularly M2 is producing the strongest, most robust rhythm, which is kind of interesting and something I'll need to keep working on.
And I'll just ask a quick question. So you showed us beautifully about the collagen structures that these cells live in, but all your experiments are 2D. So have you done anything to try and model that 3D environment, see if that changes some of these results?
No, but yeah, that's a really good point, because I think there is lots of studies to show that the collagen kind of fibril formation isn't just linear. Obviously, it goes between the cells, so it would be good to see, what happens if you make it into a 3D format.
Okay. Oh, Dan has a question.
So I think cardiac fibroblasts, macrophages in particular produce, can produce lots of collagen, as their fibroblast counterparts do. But yeah, I haven't looked into that yet, but it's a good point to look at actual different types. Yeah, I think so. Yeah, initially I thought it was just going to be a simple protein, but then after the heat treatment, and it still induced a rhythm, I think it's going to be more complicated, probably, as you said, a kind of conglomerate of protein, I'm not sure. But we also need to work out whether it's just a protein with really strong disulfide bonds by using reducing agents and stuff like that. So yeah, I think there's a lot more to do.
Okay, if there's no more questions, just thanks again, Katherine, for a lovely talk.